All right, today we're going to dive into a technique that's basically a secret decoder ring for the human body. It's called H&E staining. We're going to see how two pretty simple dyes can take a piece of brain tissue that's essentially invisible and just turn it into this stunning, completely readable map.
So let's just start with the big question, right? How do scientists even begin to study the incredibly complex architecture of the brain when a thin slice of it, say from the cerebellum, is almost totally transparent under a microscope?
Now, before we get to the solution, you really have to grasp the problem here. Imagine you're looking at a piece of tissue under a really powerful microscope, and you see, well, almost nothing. It's a world without color, where all these complex, important structures are just invisible. I mean, think about it. It's like trying to read a book with completely blank pages. That's really the problem we're facing. You might see the outline of the tissue, but all the crucial stuff, the different cell types, the distinct layers that each have their own job, it's all just washed out, a total mystery. So, how do you actually write on those blank pages? Well, you need ink, right? And in the world of histology, their ink is stains. So let's meet the heroes of our story, the dynamic duo that completely solves this problem of invisibility, hematoxylin and eosin. Now, these two dyes, they're like a perfect team. Hematoxylin is what we call a basic dye, so it's attracted to acidic things. That means it goes straight for the cell's nucleus, which is packed with acidic DNA, and stains it a deep blue or purple. You can think of it as highlighting the cell's command center. Then you've got eosin. It's the total opposite. It's an acidic dye, so it binds to the basic stuff, like the proteins in the cytoplasm. It colors in all the machinery around that command center in all these shades of pink and red. And that brings us to this key term you see here, basophilic. It sounds kind of complicated, but it just means base-loving. The acidic DNA in the nucleus basically acts like a magnet for the basic hematoxylin dye. It's this simple chemical attraction that's the real secret behind why this whole thing works so beautifully. Okay, so we've got our dyes, we know what they're targeting, so how do we actually apply them? Well, it's not just a simple dip in a bucket. It's a really carefully orchestrated process, almost like a staining symphony that brings the hidden structures of the tissue to life. Now, the real lab procedure has like a dozen or more steps, but it really boils down to these four critical stages. First, you've got to prep the tissue and rehydrate it. Then comes the blue bath. This is where hematoxylin does its thing, latching onto all the nuclei. After a quick rinse, the slide gets its pink counterstain from the eosin, coloring basically everything else. And finally, the finishing touches. The tissue gets dehydrated and cleared, making it transparent again, but this time with all those vibrant colors perfectly locked in place. All right, we've set the stage, we've met our dynamic duo, and we've walked through the whole process. Now, it's time for the big reveal, the payoff. Let's see the incredible transformation this staining symphony really creates. Just remember, this is where we started. A ghost. A formless slice of tissue, full of secrets, but revealing absolutely none of them. And boom, here is the result. Just look at that. It's not a mystery anymore, it's a map. The blue and pink dyes have uncovered this hidden world, a complex, beautiful architecture made of distinct, functional layers. And this is the incredible part, right? We can actually read this map now. See that pale pink area up top with just a few blue dots? That's the molecular layer. Right below it, you see that neat single line of large dark blue cells? That marks the critical Purkinje cell layer. Then you get to that super dense dark blue region. That's the granular layer, just packed with cells. And finally, the light pink region at the bottom is the white matter, which is like the brain's information highway. Every single color and pattern tells a story. So this is absolutely beautiful to look at, no question. But its real power, well, it isn't just about making pretty pictures. This technique, which is over 100 years old, is a frontline tool in modern medicine that literally helps save lives. And it's really no exaggeration to call it the gold standard. I mean, for any pathologist looking at a tissue sample from a biopsy, this is almost always the first and most crucial step in figuring out what's going on. And why is that? Because it's fast, it's cheap, and it is incredibly reliable. Just let that sink in for a second. For over a century, with all of our amazing technological advances, nothing has managed to replace this simple, elegant solution. It stayed indispensable because nothing else gives you such a clear, immediate overview of tissue architecture for so little cost and effort. So, when a pathologist peers at one of these slides, what are they actually looking for? Well, they're like detectives on a case, searching for clues. Are critical cells, like the Purkinje cells, missing or damaged? That could signal a specific neurodegenerative disease. Is the layering all messed up? Or are the cells themselves looking weird and disorganized? That could be a telltale sign of a tumor. Because at the end of the day, these patterns of blue and pink, they're not just a picture, they're a language that lets doctors read the health of our bodies at a cellular level, telling a story that can diagnose all kinds of things, from brain injuries to cancer, and help guide life-saving treatments. You know, this whole process really leaves you with a fascinating thought, doesn't it? H&E staining proves that a massive amount of information can be hidden right in front of our eyes, completely invisible, until we find the right way to look at it. It just makes you wonder, what other secrets are out there, hiding in plain sight, just waiting for the right colors to bring them to light?
Welcome. Today we'll explore the fascinating world of cerebellar tissue staining, a critical process in histology that helps us visualize the intricate structures of the brain. The main purpose of this staining technique is to highlight different cellular components, making it easier to identify normal and abnormal features under the microscope. Chemistry behind this method involves two key dyes: hematoxylin, which binds to basophilic acidic structures like nuclei, and eosin, an acidic dye that stains cytoplasmic and extracellular proteins. Together, these stains provide a vivid contrast, allowing for detailed examination of cerebellar layers and cellular architecture. Understanding this chemistry is essential for accurate tissue interpretation and diagnosis. Let's walk through the step-by-step procedure for staining cerebellar tissue. First, the tissue is rinsed in 1x PBS to remove debris and balance the pH. Next, it's washed in tap water to rehydrate the sample. The tissue is then stained in Mayer's hematoxylin for about 30 seconds, which colors the nuclei. After another tap water wash to remove excess dye, the nuclei are blued in 1x PBS for 20 seconds, converting them to a crisp blue shade. Hydration follows, using 70% and then 95% ethanol. The tissue is counterstained in alcoholic eosin for 30 seconds to highlight the cytoplasm. Further dehydration through 95% and 100% ethanol stabilizes the tissue and removes water. Finally, the sample is cleared through 3 changes of xylene, making it transparent and ready for cover slipping. Each step is crucial for optimal staining results. Once stained, the cerebellar tissue reveals its distinct layers under the microscope: the molecular layer, Purkinje cell layer, granular layer, and white matter. At higher magnification, you can observe the unique features of each layer. The molecular layer appears less dense, while the Purkinje cell layer is marked by large flask-shaped neurons. The granular layer is densely packed with small cells, and the white matter shows up as lighter regions. Recognizing these features is essential for identifying normal cerebellar architecture and detecting pathological changes. This clear differentiation is what makes staining such a powerful diagnostic tool in neuropathology. Interpreting cerebellar tissue staining involves looking for both normal and abnormal patterns. Common pathological findings include cerebellar degeneration, characterized by the loss of Purkinje cells, gliosis with increased glial cells, demyelination seen as pale, eosin-deficient white matter, and tumor identification, which distorts normal layering. Sometimes technical issues arise, such as faded nuclear staining, muddy or gray nuclei, or weak eosin staining. These problems are often caused by overdifferentiation or weak hematoxylin. The solution is straightforward: use fresh hematoxylin or extend the staining time. Mastering both interpretation and troubleshooting ensures reliable and informative results for diagnosis. Welcome to